science fans! Welcome to SHE-ensya. Our topic for today is the scientific method. The scientific method is a set of processes that has characterized the natural sciences since the 17th century. It involves experimentation, observation, and the testing of various hypotheses. The scientific method is the backbone of research, and it is important for us to understand how it works. A lot of people think that the scientific method applies only to scientists. Actually, if we employ the scientific method to our daily lives, we would be less vulnerable to alternative facts and fake news. We would know which information to trust and which information we should avoid. So learning about the scientific method is not just essential for scientists, but to basically everybody in the world. The first step of the scientific method is to make observations. For example, in the recent outbreak of COVID-19, we know that the disease is being caused by a virus that is part of the coronavirus family, specifically those involved in creating the severe acute respiratory syndrome. The first set of outbreaks occurred in Wuhan, China last December 2019, and it has spread around the globe since then. As of the making of this video, there are already 202 cases in the Philippines with a fatality of 17. There has been more than 200,000 cases worldwide, and the global death toll has already reached more than 8,000. Antibiotics do not work against COVID-19. Just like in any viral infection, we have to rely on our own immune system in fighting against the virus. And since it's a new virus, our immune systems are having a hard time recognizing it and fighting back. Topmost in the minds of people is if a vaccine can actually work against COVID-19. There is already a vaccine that works against influenza. So can a vaccine really be used against this new disease? The next step in the scientific method is to consult prior knowledge. What is already known about the topic that we want to learn about? And in the case of vaccines, if we look at the large family of coronaviruses, despite 17 years from the first recording of SARS and MERS, no coronavirus vaccine has yet to be created. This is mostly because of the rapid rate of evolution or mutation that the virus undergoes. It is a very challenging procedure for us to develop a vaccine that targets something that changes all the time. But this doesn't mean that there is no hope. We have collected a large volume of information about the prior coronavirus outbreaks. And because of all these prior knowledge, it is possible to find something that is common across all of them and design a vaccine. So we move on to the next step of the scientific method, which is the formulation of a hypothesis. This aspect of the scientific method is challenging to most students and scientists. And so before we move on to the process that we are studying, let me first define what a hypothesis is. A hypothesis is a tentative, falsifiable explanation of an observation that can be tested using experimentation. One of the common experiments we have in our ecology classes at the De La Salle University is to check which glassware is best used to measure out 30 ml of water. You can see in my background 3 different types of glassware, and we ask them to formulate a hypothesis based on that. There is a huge difference, however, between a hypothesis and a prediction. Most students would say that the pipette would be most accurate, and that is a prediction. A hypothesis, on the other hand, is an explanation. In this case, a possible hypothesis could be that the glassware with the smaller diameter could be more accurate compared to the others because the meniscus would be more accurate and easily identifiable. So that is the difference between a hypothesis and a prediction. A A hypothesis should be based on prior knowledge. It is, after all, an intelligent guess. So, for the case of the development of the vaccine for COVID-19, common hypothesis in the papers that I've gone through states that the spike S protein of SARS-CoV is the major inducer of neutralizing antibodies, and the receptor binding domain, or RBD, in the S1 subunit of S protein contains multiple conformational neutralizing epitopes. This means that the active portion of the SARS-CoV-2 virus is in the RBD domain. This suggests that the proteins present in the RBD domains and those that encode the sequence for this portion can be used to develop the safe and effective SARS vaccine. What this implies is we must target the portion that is most active in causing the disease. So if you look at the structure of the SARS-CoV-2 virus, we would want to target the RBD portion. The next step of the scientific method is the collection and analysis of data. This would involve a lot of experimentation. And in the case of the vaccine for COVID-19, human testing has begun in Seattle. The first patient has already been injected with the vaccine that was created, and her reactions to it are being observed and recorded. Whenever we undergo human experimentations, we have to remember the three core principles of the Belmont Report. The first is in autonomy. The persons who are involved in the research must have a clear idea of what the research is for and the risks that they are to undergo. The second is in beneficence. The experiments that they are undergoing should be for the greater good. And lastly, justice. Everyone should have equal advantages in the experiment that is being undergone, and everybody should share on equal responsibilities as well as benefits in the pursuit of the research. When it comes to experimentation, there are certain experiments that are better than others, and it is all dependent on the design. So there are certain key aspects of the experimental design that you must take into mind. First is sample size, which refers to the number of subjects in a group that you are going to test. Limitations on this could be your budget, the time that you have, but generally, the greater the sample size, the more consistent your experiment will be. Second would be your independent variable. What in the experiment are you manipulating in order to check its relationship to the dependent variable, which is what you are measuring? For example, whenever we're testing vaccines, the independent variable, or what we're manipulating, could be the titer or the concentration of the vaccine that is injected into the person. And the dependent variable is how much reaction from the immune system of the person are we getting. Next, you also have a standardized variable that is held constant for all subjects. So for example, all individuals that the vaccine could be tested for should be of a particular age or of a particular sex. This is so that we can test for the different reactions of people across different age groups to a vaccine or across the different sexes. Finally, you should also have your control. In human experiments, sometimes the control group is called the placebo group. You don't know if you are being tested the actual vaccine or medicine, or if you're just being given a placebo like a sugar pill or an injection of a saline solution. This is to prevent any psychosomatic response from contaminating or interfering with the data. Once all the data has been collected, the next step would be checking for statistical significance. This is to ensure that the data that we see or the trends that we observe is caused by an actual correlation or connection between the parameters that we're testing and not just because of coincidence. Once we have all the data with us again, we go back to prior knowledge. We check the different trends on the reaction of the immune system to existing vaccines and if they match the new vaccine or not. We look at the length of protection that different vaccines has given to people so that we can estimate or approximate how long the new COVID-19 vaccine will be useful. Prior knowledge will help us fully understand the implications of the data that we collected. For a lot of scientists, this portion might be the most euphoric or the portion that makes us the most happy, the Eureka Moment or the conclusion. We will know whether we are successful or not, and therefore we can plan on the next steps that we're supposed to do. However, this happy moment is very much short-lived because there are still a couple of steps left. The next step in the scientific method is peer review and publication. We have to show our work to fellow scientists and they would check it to see if we were able to do the science right. This ensures that as scientists, we perform the experiments with integrity and without bias. This also minimizes and prevents people lying about their work and pretending to have created something that is beneficial to mankind. This can be a very painful process because you're opening yourself up to criticism. But at the end of it, the scientific method and criticism is the backbone of good science. A common misconception in science is the difference between a hypothesis and a theory. A theory is actually a pretty big deal because it is of much greater scope than a hypothesis because it is backed by a large body of observations. Despite this, however, a theory is not a fact. Just like a hypothesis, a theory is also falsifiable. A fact, on the other hand, is something that is consistently observed and has already undergone a lot of testing. And because of its consistency, it is considered to be true at all times. So, whether we're looking at the COVID-19 vaccine or to see whether or not the cake in the newly opened restaurant around the block is really good, the scientific method can help you make decisions that are based on facts and not feelings. This lessens the bias and prevents you from making irrational decisions about certain topics. So let's go back to the different steps of the scientific method. First, you make observations and ask questions. Next, you consult prior knowledge and formulate a hypothesis. You collect and interpret data as many times as you possibly can. You consult prior knowledge again and happily conclude. And finally, ideally, you undergo peer review and publish your research. So, I hope you found our short discussion on the scientific method helpful and useful for you. If you want to see more of this content, please let me, your resident Filipino scientist, know about your thoughts in the comments section below. Please like this video and subscribe to my channel also. Remember, on doubt, always use your conscience. Thank you so much and see you around! Hello, science fans! Holy cat! There's a cat in the background!